Good afternoon. Okay, we are start our second lecture. Lecture about definition of the concept of gene polymorphism and their role in the development of disease. It is topic about our second module in our course. In first course, we speak about protein polymorphism, how it have development, and go to gene level. Our second module will about gene and polymorphism gene, type of polymorphism gene, and you in your topic will continuous make report in your group, use all your previously report and materials. Key terms. Gene polymorphism. This is terms was given Martin Kreitman in 1983. It is the multiplicity of the manifestation of genes due to the existence of allelic variants of genes. Predictor genes. This term was given Daniel Nobert, 1997, and say about propensity genes, that is allelic variants of genes or polymorphisms that contribute to development of disease. Next, multifactorial disease. This term was given Lionel Penrose, 1947, and say about diseases in the emergence and development of which lies the cumulative effect of exa and endogenous factors. We always can to meet polymorphism in our usual life. And if you will to see for these pictures, you can to see many different color of these birds. The occurrence of two or more different forms or morphs in the population of a species is referred to as polymorphism. What is meant by polymorphism in biology? In simplest terms, polymorphism is any process. There are two or more possibility of a trait are found on one gene. This is give information if you make Google and find first info about what means polymorphism. And real, if we will look for some cell, for our classmate, for our relatives, we will to see many same but little other shape of nose, other shape of fingers, of structure hairs and other. And all this give to us uh, polymorphism. And again, I return to our first model and will say about polymorphism of proteins or enzymes give or basis on polymorphism of genes. Sometimes we now know about other mechanism, but also present polymorphism genes and variation in genetic materials can lead to variation in phenotypic. And again, look in these pictures and what I said about, you can to see here all same but always present different morphs which we say about individuals, groups and population. Many, little, some. Polymorphism has caused controversy about its role in evolution. But if it essentially follows a natural evolution, it serves as a reference in contrast for the study of natural selection. It is also used by ecologists in the conservation biology to reconstruct the past history of species. Polymorphism and mutation. It is a process, it is one way. Mutation may be due a change from one type of nucleotide to another, an insertion or a deletion or a rearrangement of nucleotides. And you can to see here in these pictures many reasons which can to lead for mutation. It is cellular metabolism, oxygen example, or any toxic things. It is light of sun, ultraviolet. It is radiation. It is chemical. It is replication errors, which always can happen. All this act on DNA and one or two or three nucleotides can has mistake. It may be insertion, go no, come to new lead, DNA lead. It can deletion, go away. It may be repeat 2, 3, 4 nucleotides many times, and many different variants. In our biology course, we speak about this, and now we need to understand if mutation will give new shape, new genetic information for proteins, it may be disease from one protein. Example is phenylketonuria. When we speak about one mutation, give one disease. But when we speak about polymorphism, we little need to think about other situation. It is will change genetic materials, but it is not will critical for reason of disease. So it will new shape, new function of enzyme example, but it is not will reason for disease. This is process mutation and appearance new variant of genetic materials and new variants of protein materials, always in evolution process, always. And in this picture you can to see 4 types independent de novo mutation. It is A, ancestral standing genetic variation. It is B, ecotype, disparial and secondary contact, C, and introgressed genetic variation, D. All this connection with mutation. Sometimes mutation make good and make easy for adaptation and for living organism with new character. Sometimes it is give lethal effect. We speak about our biology class about lethal mutation, lethality mutation. Sometimes it is only make new features, new possibility for adaptation. All mutation in this way can divide it for some groups. It is neutral, harmful, or advantageous. And previously slide I explained this is neutral, harmful, or advantageous effects. Also, we need to see on this slide and need to understand all this evolution process. It is way of adaptation. Yes, mutation can happens, but it gives different possibility for all species which present and which has new mutation. And different area, it may be different situation with neutral or harmful or damage mutation. And in this picture, blue and red can to show you how it can to distribution in some area. Sometimes we can to see together 1 and 2 species of fly. Sometimes we can to see only red and sometimes only blue. And in picture right down side, you can to see little road, blue species with a little river, and here present fly with short wings. They can fly for short distance and all times stop if it will need or necessary. Right side we can to see red fly, and here you can to see wings very big, and here river or maybe lake very big. So this is fly need make action long time, and this is a shape of their wings can help to fly from this big area. And we understand that if we change fly from one side to other side, uh, blue fly with short wings can die because they cannot fly through this big distance which present on lake. Uh, It was previously was about animal, yes, insect, but now you can to see information about human evolution, and this is map present many different character which has only Y chromosomes of human, and how it is distribution for all continents, and you can see population with more same different genes and isolation one area from other. All this happens in evolution, and let's when million years ago people come from Africa and need to adaptation for different condition around condition environmental which they met on his volcanic. And in evolution theory, which has now scientific evidence, we need to think about over the past 50, 100,000 years, Homo sapiens has settled across the globe, associated with a change in the climatic zones to temperature and Arctic. 40 from 30 till 40,000 years ago, a modern type man penetrated the subarctic and Arctic zones of Eurasia. And more adaptive need to climates. And I hope that all you can to come in Russia, in Simferopol, and let's to study in our country, and you can to use your adaptive systems. You can understand how it is adaptation work for different climate, and you can note who was adaptation fast, who need more time for this reaction. Genes which involved in adaptive process, more it is genes which need to adaptative for cold resistance. And these genes includes thermoregulation, response to temperature stress, energy metabolism, lipid metabolism, regulation of muscle contraction, regulation of blood pressure, signaling and cell-to-cell interactions. And also you can to see here how was distribution human population from one place, Africa, to other place of continent. And this is give that all population has different mutation which can give more maximal and positive adaptation. And it lets that small amount of allele which have more good effect can distribution in population from 0 to 30–50%. And more good it is when they was in heterozygotes and all times present in populations. No, also you can to read about different mutation. It is all about from evolution genetics. And for us, need to understand that all mutation can use as a function. We know that one amino acids can code three nucleotides, genetic code. We also speak about this in medical biology class. And it's give for us information that genetic code or mRNA always present as leads with nucleotides A, T, G, C, and so on, different combination. But it is give triplet, 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 1 from 2, from 3, from 4, from 4, 5, and so on. And it's make coding amino acids leads, which give our final proteins. And if change one letters or one nucleotides, add or delete or repeat, all our genetic codes will change and amino acids leads will new, will other. But also we remember that it is not need 100% that will other, because we remember that combination of triplet present 46 and present also stop and start codons, but it is a small amount. But we know about amino acids only 20, so many different triplets can coding one amino acids. And so if polymorphism or mutation will lead to in one triplet, one nucleotides will change for other and not change amino acids, we can to say about synonymous polymorphism. And if change, so we will speak about non-synonymous substitution result in an alteration of the encoded amino acids. And here we also can to say about two types. It is missense mutations, when happens change the protein by causing a change in codon, and nonsense is a result of misplaced termination codon. And example, if it is proteins, they will more short. Now, also here I give you information that how tree, it is genetic tree, how happens not only the geography structure of human genetic diversity, but how genes can make clusters, family. When we speak about protein polymorphism, we also say about protein families. And here also we can to see genes families. They was one, they try from left side we see one genetic information which coding one proteins, then in adaptation or other mutation give many different shape, polymorphism, and foundation. This is family of genes which can different in other continent, different in other nationality, population, and so on. And when we speak about polymorphism, more, we will speak about SNPs, CNP, single nucleotide polymorphisms. And if now all people know about 51 populations, and this is 51 population, scientific and base date has information about 650,000 SNPs, which give only 11% differences interpopulation. 36 populations, 32 from Eurasia and 4 from HapMap, was analyzed in genome project, in polymorphism project, 200,000 SNPs, and find 13% diversification. In Pan-Asian CNP consortium was find in Asia population differences only 6%. It is little part, but this is little part near 100,000 SNPs, and every of them can lead to disease. One can to lead to disease, 2, 3, combination from 4, from 5, and differences. So it is global differences between people, and so differences reaction for adaptation, for pathogenesis of disease, and other. And this slide, you can see main type of polymorphic variants. And I take for you only, this is more famous for, it is restriction fragment length polymorphism. It is variable number tandem repeat and short tandem repeats and single nucleotide polymorphisms. And from label, from name, you understand that we need to speak about copy number polymorphism variation. When nucleotides leads have a repeat short or more big. And in these pictures, you can also to see that different people can have different repeats, different this is position. What means tandem repeat polymorphism? In this slide, we will speak about short tandem repeat. Short. 3 nucleotides in right part of our picture repeats in man 1 5 times, in man 2 6 times, in man 3 7 times, and in left part of our slide you can to see SNP. And here only one nucleotide will change in two cases, man1, comparative man2 and man3. But this is enough for make little different function of proteins or enzyme. Also, when we speak about polymorphism, we need to understand where foundation this is polymorphism. And in this slide you can to see many different position or many different part in our genetic in our chromosome or DNA. It may be genes, it may be intergenetic space, it may be introns, and it may be exons. So not all polymorphism can have new effect. It is maybe part non-coding, and it is not will make any new effect. Also, tandem repeats can to divide it for some groups. It is satellites. When we speak about tandem repeats, we speak about satellites. And satellites, it did repeat from 1 to some 1,000 base pair in DNA. And here we can divide it for mini satellites and microsatellites. Mini satellites, it is more than 10 base pair, and microsatellites, it is from 1 till 10 base pair. All this you also need to understand because your second module in our big data practical class will about analysis which genetic polymorphism you will find in your group, in your proteins. Example, hemoglobin, which type of proteins you will find and which you will describe, and how it needs to interpreter, and how many types of different polymorphism present in one proteins. It is also different and four which need to find, which need to understand. But for you as for future doctors, main here to understand how it can use in medical scientific. What helpful understanding about type of polymorphism will for surgery or therapeutic or gynecologist or oculist. Which info need for doctor? Yes? In these pictures you can to see again tandem repeat polymorphism, and here you can to see more big differences, and also you can to see that one person can have not always same polymorphism. What it means? Individual number 3. Here present chromosome 2 copy 1, chromosome 2 copy 2. What is it? It is means that one chromosomes copy 1 example from mother, and second chromosomes copy second from father. And both parents give different variation. Different variation of this is polymorphism, but it is heterozygote condition, and it may be effect, as we speak about previously, codomination. but maybe effect as dominant, complete dominant. Yes? If we will see individual 3 as mother and individual 4 as father, so we can understand that both parents can give many combinative for this generation, for this son or a daughter. So, short tandem repeats are very short stretches of DNA that are repeated back to back at various locations throughout of human genome. Typically, the repeating sequence is just 2, 3, or 4 base pair in length, and the number of copies found back to back is variable across a wide range. Unlike the case for the DNA sequences of coding genes, there is no correct number of repeats for any specific short tandem repeats in the genome. They are simply areas within the genome in which variation is normal and healthy. For any specific short tandem repeats, each person will have 2 copies, and that is inherited from mother at concept and other that is inherited from father. Short tandem repeats are helpful in forensic and paternity testing. And main research area of research where it can use it is determination father, or example search enemy when X or Y chromosomes have short tandem repeats markers and can give many interesting info which will repeat and which will individual and which can give evidence for searcher aim. And again, I will speak about single nucleotide polymorphism. Is present at a particular nucleotide site. The DNA molecules in the population often differ in the identity of the nucleotide pair that occupies the site. For example, some DNA molecules in the same population may have a TA base pair at a particular nucleotide site, where other DNA molecules in the same population may have a CG base pair at the same site. And also in your future work, in module second, when you will prepare a report about polymorphism of genes, one of your tasks will find population differences. Example, how this is polymorphism present in Russian population, or how this polymorphism present in Indian population, and present or not present interpopulation differences. It is very interesting because in India, same as in Russia, present many population, and they 100% has polymorphism genes. And if you will find this is info, I think it will interesting for you because you will understanding many biological process, many physiological process, and many way of pathogeneity. One people have disease, but other people also eat this is food, live in this is area, but not have disease. Now again, I give for you this is information, pictures information, and also I use 2 individual shape and one individual with in upper has chromosome 2 copy 1 and chromosome 2 copy 2 from both their parents, from mother and from father. And you can to see here polymorphism has differences. Every chromosomes has a different situation. And example, individual second in his chromosome 2 copy 1 and copy 2 present same polymorphism and will homozygotes, but previously individual from one will heterozygotes. In next, when we determine polymorphism in our class, we will see only in one variant, T or A, look at chromosomes copy 1, only one variant, because we remember from medical biology that DNA has matrix, mother lead, main lead, which given for proteins transcription, and has complementary, which only close DNA double structure, but anti-complementary. So here in individual one you can to see 4 types of nucleotides, all differences, but we will in future speak only about 2 heterozygotes, example TC, and individual 2, it is homozygote CC. Understand? And also statistic information. The word allele is in quotation marks above because the CNP need not be in a coding sequence or even in a gene. In the human genome, any 2 randomly chosen DNA molecules are likely to differ at about 1 CNP site every 1,000 base pair in non-coding DNA and at about 1 CNP site every 3,000 base pair in protein coding DNA. The definition of a CNP SNP that stipulates that DNA molecules must differ at a nucleotide site includes rare genetic variation of the short found in the less than 1% of the DNA molecules in a population. SNPs are the most common form of genetic differences among people. And in our class, we more part will speak about nucleotides because all today molecular medicine, personalized medicine work as determine polymorphism, make panel of 2, 3, 5 key polymorphism which can lead to disease or which can determine effect of treatment or make prognosis of risk. And also I give a new shape of how look single nucleotides, and I want that you understand that again polymorphism, it is not mutation. It is not same as phenylketonuria. When we speak about change of nucleotides will give disease. No, single nucleotide polymorphism give only other information, but it is not pathogenity. It is only a little other possibility, but not critical. And in this moment, we need, I'm return. You will in break time to see this is pictures, understanding here, and we make break time 10 minutes, and in 13 we return and will continue our lecture. And we again start our lecture. And now you can to see restriction fragment length polymorphism. And here we can to see how need find and how need laboratory make and how then understanding and analysis this is SNPs. You see in down left part of this slide, Escherichia coli. Escherichia coli echo I. It is enzyme restriction enzyme, which can recognize 3-4 nucleotides and can make disconnection. And main here that every from bacterial enzyme, restriction enzyme, can to know only special nucleotides LEDs, only 3-4. So if we know which polymorphism we have, we can to find restriction enzyme and give him task, make restriction. Then we will make electrophoresis because amplification fragments, restriction and not restriction, will have different size, and when we will make electrophoresis, will move with different speed. More big will go from left side in this picture for right, more slowly. Small part will go fast. Down part, down, third line from down, it is molecular marker. They have many different parts with different size, and every size we know how many size, how many pair nucleotides present this. And so when we make our experience, we can to make together electrophoresis, then look level, how they moving, and determine also size of these fragments. In class, we will speak more about this and we'll try to make this and we'll try to determine. But now I want to speak about role of polymorphism in disease. Here I need to understand if this is polymorphism not give as mutation genes reason for disease, so it will interesting for development of disease in other side. And here we need to speak about polygenetic effect of multifactorial disease, where many factors can lead to disease, but genetic variation will make base for this is multifactorial continuums for development of disease. And method how can to determine this is polymorphism will basis genetic molecular biology method. It is real-time polymerase chain reaction. It is a polymerase in chain reaction with electrophoresis restriction analysis, which we before see pictures. It is electrophoresis after polymerase chain reaction. And it is a special sequence when we can to know all LEDs and find CNP. And if speak about differences and plus and minus of this whole method, uh, we need to note that sequences will more good because sequences led to determine or find new SNPs, but polymerase chain reaction give effect of finding only when we know about polymorphism, because need use special enzyme and restrict enzyme, and if we not know about polymorphism we cannot to find, but sequences make one nucleotide determine next, and so can find new and many new SNPs. How it used in a human population. In this picture, you can to see case control study. In left side, in round, present healthy donors. It is a control group. In right side, in rounds, present patients with duodenal ulcer. And all this patients and control members group persons present any genotype, any polymorphic variant or SNPs. In healthy donors, we can find AB, AA, BB, so present homozygotes AA, a wild type of nucleotides in 60%, heterozygotes AB 30%, and BB homozygotes, this is nucleotides, mutant nucleotides only in 10% case. In patients group, we can to find also all variants, but wild type of nucleotides in homozygote conditions we can to find only 20% patients. Heterozygotes same as in control group healthy donors and patients also. And mutant group present 50, one half of patients group present mutant genotype, and we need look common this is distribution of genotype, need to understand maybe this is mutant allele in homozygote condition can to lead to disease. And what we need to meet, what we need to make, we need to determine statistical evidence. And really it is work, and this is analysis we label as criteria the relative risk RR and the odds ratio OR, or the strength of the associated of the DNA markers with the disease are calculated mathematically. It means that if genotype big in patients groups, it is not 100%, it is not need must leads or will case with disease, associating with disease. So we need to find this is mathematical evidence for understanding this. And here in this slide you can to see this is mathematically reasons. How will search? BB it is main which we need to understand associated or not associated with disease, and how many in healthy 10%, how many in patients 50%. All other other wild variant of uh, normal allele and heterozygotes, we combine and give how many also in this table. In healthy, this combine of wild type of allelic can to meet in healthy group 90% and in patients group 50. And then you can to see formula which use for understanding how many differences. And we in our sum let's that OR это odds ratio will more than 1. If more than 1, so it means this is connection with disease. And if we will find BB genotype in patients, we can to think, aha, it will disease, or now or very soon. Next information about genes, which you will use in our module second, and which you will find in database. It is position of this is polymorphism in chromosomal map. And here you can to see map from not all chromosomes, only 1, 2, 4, 5, 6, 7. And you can to find P and Q and band and heterochromatin and euchromatin and location sum of genes. Example, chromosome number 1, it is first left pictures, present location of interleukin 10, Q31, Q33. It is address of this is gene. Also, we use in our class when we speak about map in biology, and I think you remember. If you not remember about this position, you need return to class number 4, if I not mistake, I think 4, and look this is info. So we can to see in on chromosome number 2 position of interleukin 1, chromosome number 4 position of interleukin 8, 2, chromosome number 5 position of interleukin 4, chromosome 6 TNF alpha or tumor necrosis factors, and chromosome 7, it is interleukin 6. All has his address. All can let location or P short arm or Q long arm. But all this is genes. It is not simple genes. It is interleukin and family of this is proteins, and they have inflammation and anti-inflammation role in pathogenesis of disease. And this is all genes make cluster of inflammations genes. And also in our module second, we will speak about cluster of genes, and you will need find cluster of genes, and you need understand that it is group of genes which can location in different chromosomes. And this is map, and this is info will determine by the coordination function of a specific group of genes function as part of a specific gene network. Because again, I will return role of polymorphism. It is make only new shape or a new amount of proteins. But altogether, they can make new big effect and need to research for one disease as a panel, same function, different genes, which give common pathogenity effects. And in multifactorial disease, it affects not the structure of the genes, but the regulatory mechanism of expressions. And questions. We have questions here. How it will work? The overwhelming majority of genetic variants or alleles of the genome are neural, indicating not so much their pathogenic role and association with disease, but rather a natural balance imbalance. At the same time, change in many genes, other regulatory regions, can not only contribute to the onset of disease, but also protect against them. And it is same. And now you can to see about different position in multifactorial disease. And here I need to understand. Now we need combine all our knowledge from first part of lecture about adaptation when mutation was happens and different person has different environmental factors which can act for this person. Also, they have different polymorphism which can take these factors and give your own adaptive reaction, and only altogether will give big pathogenic effect. And also here I present database. Some of them we will use in our class. And here you can to see and can to find between our class genotype. And this is maybe it will NCBI, which you use for proteins, and which info and which file you can use. And also I want to say about our class will in this Saturday, because we will work not in Monday, we will work on Saturday. It is information only for division 1. And thank you for your attention and goodbye.